Hello everyone. In this video we'll talk about bioinformatics and this is a Q&A session. This is the third Q&A session and the previous two are linked in the I button. This particular Q&A session is dedicated to GATB 2021 because in GATB 2021 bioinformatics is a new topic. So there are three new topics: bioinformatics, bioengineering and animal and plant tissue culture methods. In this video, we'll focus on bioinformatics, and in other videos, I have already covered other two topics. Don't forget to check out all the playlist. But after analysis of previous year question and identifying the potential hotspot, I've came up with 10 questions which would take 20 minutes of your time. So stay tuned till the end of this video. These 10 questions might give you a very deep insight that what you might expect in the exam. But before that, let me quickly tell about some important announcements. If you are preparing for MSc level or INT PhD level entrance examination, or even let's say JEE or NEET level entrance examination, Unacademy could be your one-step destination because Unacademy is providing a lot of scholarship. One such scholarship test is going to happen on 14th or 15th August, and the overall prize is worth 11 crores. This is for IIT JEE and NEET UG aspirants. This is the detailed schedule for that. Now, if you become a Prodigy and give this Prodigy mock test and you can secure top 2 ranks, then you can get your first year medical college fee totally sponsored. Okay, if you are a good rank holder, let's say if you're from 1 to 10, you can get a MacBook. Other rank holders can also get very nice gifts. Also, the rank holders in this admission test can get scholarship benefits. Like you can get 100% scholarship if you score within a 1 to 10 rank. You can use my code AP10 to unlock this and give this test. If you know any friend, any brother or sister, do share this. It would be really beneficial for them. Not only that, when you take Unacademy subscription and you participate in the weekly quizzes and daily practice sessions, one lucky winner can win an iPad daily, right? And last year, these are the list of prodigies who scored nicely in this test, and in the final test, they have ranked high. So you can share it with your brother, sister, or whomever you know who is giving IIT JEE or NEET. You can use my AP10 for any Unacademy category to get a 10% discount. Let's get back to the question answer session. So question 1 says identify the incorrect statement regarding BLAST. Here are the 4 statements. One commonly used scoring matrix for BLAST is Blossom 62. BLAST can be performed for protein as well as DNA sequences. Following the discovery of a previously unknown gene in gorilla, a geneticist will typically perform a BLAST search against the human genome to see whether it matches with the human gene. And lastly, BLAST is a global alignment tool. Let me quickly tell you that BLAST is a local alignment tool. It's not a global alignment tool. So this is a critical point that you need to remember. Other than that, all these 3 points regarding BLAST is true. The scoring matrix is Blossom 62. BLAST can be done for nucleotide. It's called nucleotide BLAST, or it could be done for protein as well, protein BLAST. Both are present. And yeah, if you get some kind of new sequence in any species, you can compare it with the known database of other animals such as human, other primates, etc. to get an idea about the homology. So that can be done with BLAST. Question number 2 says choose the correct statement. Statement 1 says in BLAST result, identity score shows which character match perfectly in our alignment, but it does not imply there is a meaningful match. Statement 2 says the E value is a parameter that describes the number of hits one can expect to see just by chance when searching a database of a given size. Both these statements are incorrect, both these statements are correct. One is correct but 2 is incorrect, 2 is correct but 1 is incorrect. So if you have guessed correctly, the correct option would be option B. Both these statements are correct, and you need to really take a note on these kind of questions. BLAST is one of the very important topic from which you can expect a question in GATB. Okay, identify the incorrect match in the following. So PDB contains structure-related data typically obtained from X-ray crystallography, NMR spectroscopy, or cryo-electron microscopy. DDBJ, it's a primary database. PFAM is a database of protein families that include their annotations and multiple sequence alignments generated using hidden Markov model. And lastly, CATH is a primary database which do not have annotations. So you have to choose the correct option, and the correct option is CATH. CATH is actually a secondary database. I have made a video on CATH. Look at it. Link would be provided in the I button. CATH CATH is a curated database. So all the sequence information from the PDB is taken into the CATH, and layers of information is added with that. Annotations are manually done. So CATH is highly annotated database. Okay, you have identified a novel protein which was not described previously. After sequencing the protein in order to predict its domain structure and overall architecture, which database you can use? DDBJ, CATH, PDB, or PIR? Yes, the correct answer is CATH. CATH can help you to predict what type of domain and what type of structure is present in your protein based on known knowledge. So CATH, if you want to learn more about CATH, you can click on I button. Question number 5, choose the correct statement. Statement 1 says, in CATH database, the architecture and hierarchy is equivalent to the fold level in SCOP. In CATH, structural hierarchy at the class level, domains are assigned accordingly to their secondary structural content, that is alpha or beta or a mixture of alpha beta. And it focuses more on secondary structures. So you have to understand whether these statements are correct or incorrect. Actually, both these statements are correct. So CATH and SCOP databases are really important for GATB. Take a note on that. Question number 5. Which one of the following is not an example of primary database? This category of question or this pro forma of question is very common. It can be asked. So NCBI, GenBank, DDBJ, EMBL, EBI, and Flybase. And you have guessed correctly, Flybase is not a primary database. It's a derived database or a secondary database. It generally takes all the information from all these primary sequence repositories and then annotate those sequence, and it has a bunch of other information as well. So question number 6 says, the bioinformatics tool used to determine motif is ProCheck, Prospect, Copia, Pattern Hunter. This kind of questions are actually really common in our GATB examination, and these names are really important. So Copia is the correct answer. Clustal Omega is bioinformatics tool that can perform multiple sequence alignment, protein domain prediction, protein 3D structure visualization, none of the above. The correct answer, you have to tell me. Question number 8. What is the source of protein structures in SCOP or in CATH? Very common question, but very important. Actually, CATH and SCOP take all the information about protein structure from protein data bank. Which statement about ProCheck is correct? ProCheck is a very important term for GATB examination. ProCheck is an important tool. So these terms are: ProCheck checks the stereochemical quality of a protein structure. ProCheck form number of postscript plots analyzing the overall residue by residue geometry. It includes ProCheck NMR for checking the quality of an NMR derived structure. And last says all of the above. So this question is kind of a quick description of ProCheck. All of these statements are correct, and this particular question gives you a very quick idea about this particular topic or ProCheck. Question number 10 says, dash alignment does not assume that the two sequences in question have similarity over the entire length. Local, global, heuristic, and cluster. So you have to answer correctly. And this question I'm leaving for you guys. Do let me know the answer in the comment section. So as usual, if you like this video, give it a quick thumbs up. Don't forget to like, share, and subscribe. Thank you.